Ciao, my fellow flora and fauna. My name is Mimi. I am a master's student in Dr. Kevin Simonin's Environmental Plant Physiology Lab here at San Francisco State University. I'm here today to talk to you about my project, tracking leaf trait differentiation of the newly diverging subspecies Chenopodium oahuensis on the Hawaiian Islands. Some things that you should know about Chenopodium oahuensis before we get started is that Chenopodium oahuensis is the only Chenopodium species to be found on the Hawaiian Islands. This means that the diverging leaf trait characteristics that we're going to be talking about today are due almost entirely to environmental pressures and not due to cross-hybridization between other Chenopodium species. Now, we sampled 6 sites across the different islands. Let's look at the map to get a closer look about what those sites look like. Here are the sites that we are sampling. All of these areas are where the plants receive direct sunlight exposure. Oahu, Molokai, and Maui are all coastal and subject to ocean seawater spray. However, Molokai is not found on the beach. Molokai population is found on sea cliffs. The last site is on the Big Island and is subalpine in habit, so it lives above the cloud line. They sometimes get snowed on even. Yeah, it snows in Hawaii. Here we have 2 images of 2 different populations on the island of Molokai, both at 4 weeks worth of growth. On the left-hand side, we have our Elio Point population, which exhibits a prostrate growth habit. So creepy crawly grows close to the ground. It has thick, succulent leaves. On the right-hand side, we have our Puka Pele site. They grow erect, so straight up and down, and you can see that the leaves are noticeably bigger. These differences in leaf morphology have led us to believe that there's also a difference in these different populations' hydraulic programming. So we're going to be doing stomatal conductance measurements, stomatal size measurements, stomatal density, leaf mass per unit area, and my favorite, Turgor loss point and osmotic potential. Let's talk more about those ones. So we measure Turgor loss point and osmotic potential by doing pressure volume curves. These pressure volume curves allow us to measure at what level of water stress each of these populations can really handle. Now, I'm going to show you a video about how this works a little bit. And basically what we do is we put a lot of pressure on this leaf until the water shoots up the reverse side of the xylem. And the xylem cells are these white bundles that you see here that form kind of this crescent moon shape. Oh, there it went. Did you see it bubble just a little bit? And now it's bubbling just a lot. So we do this measurement over and over again, once when they're in juvenile stage and once when they're at an adult stage, to really figure out, okay, how much stress can the leaves handle? How much stress can the plant handle? How much stress can the whole population handle in an ideal water environment? As global climate continues to shift, average rainfall, temperatures, and other abiotic conditions, we will see more speciation events like this one. Chenopodium oahuensis is adapting to new climates and new environments. But with rising sea levels, some of these coastal sites are in danger of degradation or disappearing completely. My research will influence the understanding of what levels of water stress that each of these Chenopodium sites can handle, increase our understanding about how the native Hawaiian Chenopodium species are responding to variations in climate, which will provide baseline information for conservation efforts, such as which populations should be prioritized given our current climate projections. We will be donating seeds that we collect at the end of the experiment to restoration organizations in Hawaii, which will directly impact other species that depend on Chenopodium oahuensis. Thank you so much for coming and listening to my talk. My plants and I thank you so much.